Hello friends, the topic for today is DNA replication. This is the first part of DNA replication that I'll be talking to you about, and the second part of DNA replication I'll be taking as a second session tomorrow. What is DNA replication? DNA replication is the process by which an identical or duplicate copy of DNA is formed from an existing DNA molecule. This occurs during cell division, that is the S phase of the cell cycle. The DNA is replicated only once during its life cycle, and this has been best studied in prokaryotes, particularly E. coli. Now, the DNA replication is semi-conservative. Now, when the unwinding of the double helix occurs, then we find that we can get two double helical daughter molecules. Now, each daughter molecule is composed of one parental strand, which you can see as the blue ribbon, and a newly synthesized strand, which you can see as the yellow ribbon. So each daughter molecule has got the one parental strand and a newly synthesized strand, which you can see as this yellow ribbon here. So this model of semi-conservative replication was proposed by Michelson and Stahl. Now, what are the requirements for replication? For replication, you require a DNA template. Now, each strand of DNA serves as a template for the synthesis of a new complementary strand. The most important enzyme is the DNA polymerase. DNA polymerase is the one which brings about the elongation of the nucleotide chain. And this is discovered by the scientist Arthur Kornberg in 1958. We require the bases, that is the deoxynucleotides like DATP, DGTP, DTTP and DZTP. In addition, we require magnesium ions and we require a primer. The primer could be either a pre-existing DNA chain or an RNA primer with a free 3' hydroxyl group. Now, as you can see here, this is the synthesis of an RNA primer, and this is the 3' hydroxyl group, and to this free 3' hydroxyl group is added the bases. In this particular case, the base which is added is GTP. Now, the enzyme which synthesizes this RNA primer are the primerases. And in E. coli, the length of the primer is around 10 to 60 nucleotides in length. Now, in some instances, the RNA polymerase also can synthesize primers. As a general rule, the primase uses ribonucleotides for incorporation into the primers. Now, once the primers are synthesized, then the process of elongation is then taken up by the DNA polymerase, which then moves in and takes over the synthetic process. Now let's look at the different types of DNA polymerases and what is their function. Now, DNA polymerase, as I told you, is very important in DNA replication. They catalyze the addition of the deoxynucleotides to the existing or the pre-existing nucleotide triphosphate. It has three very important properties which you can remember as the three P's. One is polymerization. What is polymerization? Polymerization means the chain elongation, process of chain elongation, or the rate at which the nucleotides are added per second during replication. Second P refers to processivity. Processivity means the number of nucleotides which are added to the nascent chain before the polymerase will disengage from the template. And thirdly, the proofreading capacity, that is, it is able to identify any errors, that is, when it is forming the complementary, forming the daughter strand or the or copying the template strand. If there are any errors, it is able to correct them. Now there are 5 types of DNA polymerases, DNA polymerase 1 to 5, but the main enzymes in the prokaryotes are DNA pol 1, DNA pol 2 and DNA pol 3. DNA pol 4 and 5 are mainly used for DNA repair mechanisms. Now these enzymes have exonuclease activity, that is, they can remove bases from either the 5' end or the 3' end, and so they are referred to as 5' 3' exonuclease activity or 3' 5' exonuclease activity. The first DNA polymerase, that is DNA polymerase 1, has both these activities. That is, it has both 5' 3' and 3' 5' exonuclease activity. The 5' 3' exonuclease activity is used in gap filling between Okazaki fragments. The DNA polymerase 2 is mostly concerned with proofreading and DNA repair. The DNA polymerase 3 is the one which has a high catalytic activity and very high processivity. That is, it is able to add on nucleotides at a very fast rate and bring about the further elongation of the primer. So its efficiency is much higher than polymerase 1, and it is utilized in the synthesis of both leading and lagging strand. But the reading of the template is not free of error. Now we have, this table gives us a comparison between the different types of polymerases, that is DNA pol 1, 2 and 3. When we look at the mass, you find that DNA polymerase 3 has the highest mass, that is 830 kilodaltons. It is able to, its turnover is around 1200 per minute. Being a large protein, naturally it has more than 10 subunits. Structural gene, the gene for polymerase 1 is known as Pol A, for polymerase 2 is known as Pol B, and for polymerase 3 it's known as Pol C. The direction of polymerization is 5' to 3' direction in case of all 3 enzymes. Now which of these has got both of these activities, that is both the 5' and the 3' exonuclease activity, is polymerase 1. It has both 5', 3' and 3' polymerase activity, exonuclease activity, whereas polymerase 2 and 3 have only the 3', 5' exonuclease activity. Processivity is, that is the number of nucleotides it keeps adding on till it gets disengaged from the template, is the highest in the case of polymerase 3. Now the role of Pol 1 is in the case is for gap filling, Pol 2 for repair, and for polymerase 3 is for replication. Now let's look at the structure of DNA polymerase 1. It is a trifunctional enzyme with 3 subunits. What are the 3 subunits? It has a small subunit of 30 kilodaltons, which carries the 5', 3' exonuclease activity, and a large subunit, which is known as the clino fragment. Mass is 70 kilodaltons, which has 3', 5' exonuclease activity and its polymerase activity. Now, what is the function of the 5', 3' exonuclease activity is that it can remove nucleotides either one at a time or as a group if there is any alteration. It helps in removing the RNA primer and it helps in synthesizing a new strand. Because it has the 3', 5' exonuclease activity, it also can proofread. Now let's look at the three phases of replication. The first phase is initiation. The replication begins at an origin and proceeds in both the directions, so it's bidirectional. So what are the steps? We have the unwinding of the two complementary DNA strands. So here we have the unwinding of the strands and the formation of a fork-like structure which is known as the replication fork. Each strand, each parental strand will then act as a template for production of a new polynucleotide strand. Now, does it start at a particular point? Yes, the particular point at which initiation occurs is known as the site of origin, that is the ORIC site. So, a unique point known as the ORIC site, and this site is around 245 base pairs long. The replication is bidirectional, and the two replication forks will meet at a point which is known as the termination site. Now, how does it recognize the point at which the replication should be initiated? There is a group of proteins known as the DNA A protein, which binds to the site of origin of replication, which is known as the ORIC site. Now, how does it bind to that particular site? It binds to that site because it recognizes certain sequences somewhere close to this region of the ORIC site. One is a tandem array of 13 base pairs, which contains a lot of uh, T. That is, you have this sequence is kind of conserved in different species of prokaryotes. So this conserved sequence of 13 base pairs constitute GAT, CT, NTT, and TTTT. Okay. Also, another conserved sequence is 4 9 base pair units, again comprising of T and A predominantly, that is TT, AT, CC, AC, A. So these sites are recognized, whether it is the 4 9 base pair units or mainly the tandem array of 13 base pairs is recognized by this DNA A protein, and then it it knows that the site of origin is close to this tandem array of bases or the 4 9 base pair units. So once it recognizes the site and binds to it, then what happens? It melts the DNA at that site. This process of melting requires the breakdown of ATP. So the initiation of DNA replication requires recognition of the origin of replication by a group of proteins that forms the pre-priming complex. What is its function? Its function is to separate the parental strand. So it unwinds the DNA helix ahead of the replication fork and forms a replication bubble with two replication forks. So this pre-priming complex includes first the DNA A protein. So the DNA A protein is binding at the site of origin or close to the site of origin, recognizing certain sequences, specific nucleotide sequences, which indicates to the DNA A protein that this is the place where DNA replication is going to start. Once it binds to that specific region, then it causes that region to melt. This melting process causes the two strands of DNA to separate ahead of the replication fork, and this process requires the hydrolysis of ATP. So this results in strand separation and forming a localized region of single-stranded DNA. Now, another group of enzymes which are involved in this pre-priming complexes are the DNA helicases. Around 9 helicases has been described in E. coli, But the three most important ones are the DNA-A, DNA-B, and DNA-C. They bring about the initial separation at the site which has been identified by the DNA-A protein. So the DNA-B protein is a major strand separator. It sort of moves into the replication fork and it kind of separates the two strands of DNA. So it's kind of forcing the two strands apart. So this is the principal helicase which is used by E. coli for the strand separation, and it is ably assisted by DNA-C protein. Then once the two strands are separated, we have the single strand binding protein or the SSB protein, which binds to the single strands and keeps the two strands of DNA separated and protects the DNA from nucleases. So the pre-priming complex has the DNA-A protein, which identifies the region near the ORIC site, binds to that region and causes the melting of the DNA and the separation of the two strands of DNA. Once the two strands are separated, the DNA-B protein kind of forces itself and forms a fork. It forms a replication fork, thereby separating the two strands of DNA, and it is ably assisted by the DNA-C protein. Once the two strands of DNA are separated, then the SSP proteins will bind to each strand and prevent the two strands from coming back together. It also protects the DNA from any degradation by nucleases. So here in this diagram we see the DNA-A protein, which is a tetramer. It is binding to certain regions which are close to the ORIC site. So these are I told you, the tandem array of base pairs that we see, which is an AT rich region. It recognizes that region and binds to that site. And once it binds to that site, then what happens? There is the melting of DNA at that site, and that is helped by means of ATP. Once there is the separation of the two strands, then you have the entry of DNA-B and DNA-C to the unbound region. Now the DNA-B is a hexameric structure and it binds to each strand and separates the DNA strand bidirectionally. Once the two strands are separated, you find that the SSB tetramers will now bind to each strand, preventing the two strands from getting coiled up again. Okay. The next set of enzymes are the topoisomerases, which will prevent supercoiling. Now there are different types of topoisomerases. You have the type 1 DNA topoisomerase and the type 2 DNA topoisomerases. Now the type 1 has got nuclease and ligase activity. It relaxes negative supercoils, whereas the type 2 topoisomerases has got both. It will prevent both positive and negative supercoiling and also separate any interlocked molecules of DNA which is formed following chromosomal replication. Then we have the DNA gyrase, which is also a type 2 topoisomerase, which is found in E. coli. So once all this is ready, that is the separation of the two strands of DNA, and this is ably helped by all the enzymes and proteins that I told you about, and especially lastly the topoisomerases, which will prevent the supercoiling. It is like a telephone wire. When you try to unwind the telephone wire, it goes back again into a coil structure. So to prevent that supercoiling, to occur, and, and to remove any torsional stress, you have the topoisomerases. So once this is set, then what happens? We have the addition of the primases. Once the primases is added to this pre-priming complex, it is then known as the primosome. And then the primosome directs the primase to form the primer, the RNA primer. Now let's look at the process of replication. Now DNA synthesis occurs in a 5' 3' direction. So that is its polarity and is semi-discontinuous. So the leading strand is the strand that is being copied in the direction of the fork movement. So here this is your leading strand and is synthesized continuously. The lagging strand is the one that is being copied in the direction away from the fork movement. So this is it is maintaining the polarity and continuously synthesizing in the 5' to 3' direction. Now the lagging strand, it cannot synthesize in this way. It has to synthesize away from the fork movement. So it is synthesized discontinuously as small fragments of DNA, and these short stretches of discontinuous DNA is termed as the Okazaki fragments. Now these Okazaki fragments are then eventually joined and ligated to form a single continuous strand, which is referred to as the lagging strand. So the next phase of replication is elongation. And the first is the synthesis. Let's look at the synthesis of the leading strand. As I told you, helicase will separate the two DNA strands. Once the two strands are separated, the SSB binding protein will bind to each strand and separate and stabilize the separated strand. The topoisomerases will be available there to relieve any torsional stress and the recoiling of the DNA. Now, once all this has been set in place, then what happens? The primer is synthesized by the primase, which is present in the primosomes. And once the primer is synthesized, the RNA primer is synthesized, the DNA polymerase 3 will now come into play. It adds the nucleotides through the free 3' hydroxyl group of the primer and keeps on adding nucleotides which are complementary or specified by the template. Elongation occurs in the 5' to 3' direction. Now, why is the DNA polymerase 3 so highly processive? Is because the beta subunit of the enzyme, it forms a ring, and that ring kind of clamps on to the template and moves along the template. It acts like a sliding clamp. So these are the events which are occurring at the replication fork. You have strand separation. The two strands are being forced to separate out by means of the DNA helicase. Any torsional stress is removed by the topoisomerases. Once the two strands are separated, the single strand binding protein will then bind and keep the two strands separate. These are the proteins and enzymes of the pre-priming complex. Then it takes on the primase. It adds on the primase, and then the primosome now directs the primase to synthesize the RNA primer. And once the RNA primer is formed, then the DNA polymerase 3 will keep on adding nucleotides. In the case of the leading strand, it continuously adds on nucleotides. The DNA polymerase kind of forms a clamp and binds to the template DNA, and thereby it becomes highly processive. That is, it can keep on adding nucleotide at a very fast pace. Now the synthesis of the lagging strand is more complicated because it has to synthesize in the 5' 5 prime 3 prime direction, but it is in a direction opposite to the fork movement. So the primase requirement is there for each fragment of DNA which is being synthesized. So each fragment will have its own RNA primer. So the primosome will direct the primase to synthesize a short RNA primer which adds on nucleotides, and then it moves to the once that is formed, then the primosome again compels the primase to synthesize again a short residue of RNA primer, and this keeps going on until the until this termination site is recognized, and then when the new Okazaki fragment is complete, the RNA primer is removed by the DNA polymerase 1 and replaced by bases but with the same enzyme, that is the DNA polymerase 1, and any gaps will be sealed by DNA ligase. Okay, so what you have to remember about the lagging strand is that it is produced as short fragments. Each fragment the primase will synthesize an RNA primer to which the nucleotides are added on to form the short Okazaki fragment. This is added on by DNA polymerase 3. Once the synthesis of the Okazaki fragment is complete, the RNA primer is removed. The primosome will then again direct the primase to synthesize another RNA primer. That once the RNA primer is formed, then again the DNA pol 3 will add on nucleotides to complete that Okazaki fragment. Okay, till the termination site is recognized. Each time the RNA primer of the previous Okazaki fragment is being removed by DNA polymerase 1 and is being replaced by bases by the same enzyme. Any gaps or nicks are sealed by DNA ligase. Now the synthesis of leading and lagging strand is coupled and is accomplished by looping of the lagging strand and concurrent synthesis by DNA polymerase 3 in concert with ribosomes. So this is again to show the structure. This is the beta subunit of the DNA polymerase 3, which acts like a ring-like structure or a clamp which clamps onto the template, and because of that it is able to add on the nucleotide at a very high rate, and that results in the very high processivity of the enzyme. So let's look at some of the terminologies that we used. A primosome is required for the repeated initiation of the formation of RNA primers ahead of the formation of the Okazaki fragment. So each Okazaki fragment, before it's formed, it will direct the primase to form a RNA primer. Okay, this primosome consists of the primase along with the other proteins and enzymes which I had mentioned, like the DNA A protein, the DNA B protein, the DNA C protein, the helicases, and the topoisomerases. All of these form the primosomes. Now, what is a replisome? So, for the simultaneous synthesis of leading and lagging strands, the replisome consists of the DNA polymerase 3, it consists of the primosome, it consists of helicases, and all the replication proteins that I mentioned. All of them form together a complex called as the replisome. This is to produce both the leading and the lagging strands together. And I told you one of the models which has been proposed for production of the lagging strand is there is a looping of the lagging strand backwards so that the orientation is in the 5 prime to 3 prime direction. The last phase is termination. So the two replication forks move in opposite directions. They meet at a site which is opposite to that of the ORIC site, and here a region is recognized which is known as the ter site or the termination region. So this region contains multiple copies of a 20 base pair sequence. Now this is recognized by certain proteins or substances known as the TUS proteins or the terminus utilization substance. And these TUS proteins will bind to this region, and when it binds to this region, what happens? The movement of the replication fork will get arrested and termination occurs. Now, what causes the separation of the daughter strands? It may be due to topoisomerase 4, but the precise mechanism is not known. So, these are the termination sequences that we see here, and these termination sequences are recognized by the TUS proteins, and they then so the termination sequences which are seen here are recognized by the TUS proteins, and they then arrest the movement of the fork, causing a termination of replication, and we have the formation of the two daughter strands. So this is just a summary of the DNA replication, which I have already mentioned earlier. Let's go on to the inhibitors of DNA replication. Now, the inhibitors are of different types. There are some inhibitors which interact directly with the DNA. That is, these inhibitors kind of place themselves between the stacked bases of the DNA complex. So it disrupts the normal structure of DNA and causes unwinding. So this inhibits the DNA from serving as a template for both replication and transcription. Examples of such inhibitors, one of the most important is actinomycin. Then you have acridine and ethidium. Mitomycin, it cross-links adenine and guanine and prevents unwinding. Now certain others are inhibitors of deoxyribonucleotide synthesis. They act at various sites, inhibiting the synthesis of purines and pyrimidines. For example, mercaptopurine, which inhibits purine synthesis, and 5-fluorouracil, which inhibits the synthesis of thymidine. Now, certain drugs which are used against bacterial infection or anti-cancer drugs can also act as inhibitors of DNA replication. Examples are hydroxyurea and sulfonamides. They inhibit also the synthesis of purines and pyrimidines. Now there is a group of compounds that inhibit DNA polymerase 3, and they are known as the aryl hydrazino-pyrimidines. There are those certain antibiotics which inhibit the enzyme gyrase, and they include norfloxacin, ciprofloxacin, nalidixic acid, and novobiocin. There are certain inhibitors of eukaryotic replication such as ethylmaldimide and inhibitors of human topoisomerases like etoposide, adriamycin, and doxorubicin. So this completes the process of replication as well as the inhibitors of replication. In my next section, I will be talking to you about the eukaryotic replication, mainly a comparison between prokaryotic and eukaryotic replication, and also I'll be talking to you about the other features of eukaryotic replication. Thank you.